Hello everyone, Brian here, and in today's video I'll be going over exercise 5-7, the citrate test. Alright, so the microbes that you'll be using for this lab exercise will be P. stewarti and E. coli. And since both bacteria are not considered BSL-2, wearing gloves will be optional for this lab. Alright, so what will you need for this lab exactly? Of course, you'll be needing your tube rack. Something different, though, is the inoculating needle instead of the inoculating loop. And that is because we don't want to over-inoculate or take too much bacteria while transferring it to the citrate test tubes because we don't want to mistake growth for just having accidentally put in too much bacteria. And of course, you'll also require your Bunsen burner. And finally, alongside that, you will need 3 citrate slants. Okay, so once you have everything ready, the first part of lab will just require you to label your citrate slants. And of course, using the little strips that have been given to you, you will label 2 of your citrate slants with the organisms that you have been given, and your last one will be your control. Now, once you have successfully labeled all of your slants, you will then want to inoculate them, excluding the control slant, as you'll want to keep that one sterile. And once you're done, just place these slants in the back of the class, and the bioprep team will then incubate them for the next 48 hours to allow for the bacteria to grow. Okay, so since I've kind of gone over the general outline of what to do in part 1, in this short little video here, I'm just going to demonstrate with these diagrams how exactly to approach it. So the first thing you're going to do is first remove the cap containing your bacteria, flame the tip over the Bunsen burner, and flame your needle. And once again, as I mentioned earlier, you don't want to take too much, so you just want to slowly bring the needle down and just kind of grab a little of the bacteria inside there, and then put the cap back on for the bacteria itself. Now, open up your citrate slant, heat fix the tip, and then transfer your inoculant onto the citrate slant itself. And remember, you don't want to puncture the surface of the citrate agar, so you want to gently just kind of slide the needle across the surface and you'll be done. And before you cap it off, you want to flame the tip again and put your cap on. However, please be careful, do not fully shut the cap or else your organism will die. So you want to make sure to just kind of close it enough so that it won't just fall off, but not all the way, so that some oxygen can come in and the bacteria can breathe. Alright, so in part 2, you'll be receiving your citrate slants, and always you want to check to see if the bacteria has grown successfully. Once you have done that, you'll then want to look for color changes, as that is a key component in being able to determine whether or not your organism was positive or negative. And of course, once you have everything recorded, you'll then want to interpret your results, and that will all be found in your lab manual. Alright, so what's going on here? When you get your citrate tests back, what you'll see is that, of course, your first test tube, which is your control, there will have been no color change and hopefully no growth either, as your control, if there were any growth or color changes, that usually will tell you that something was contaminated. So there's nothing there and no color change, then that is a good sign for your control. Now your second test tube, you'll have noticed that the color has changed to this dark blue color. This is a good sign. This means that the organism that was growing on this land here, if you look up close, you'll notice that there were some small colonies that had developed, and the colonies, of course, producing these basic nitrogenous compounds, was then able to alter the pH, making it more basic, allowing the indicator to then change colors. So if you see this color change here, that's a good sign. It means that this organism is positive for citrase. Now for your third tube, what you'll have noticed is that there was no color change at all. And unfortunately, the organism was not able to survive on the citrate medium, and thus that citrate slant will be considered a negative result. However, you do want to be careful here because the color change itself may not officially tell the whole story. And that is why you always have to make sure to check for growth as well. Because on some occasions, even though there was no color change, there can still be growth on the medium. And even in this situation, the organism will still be considered a positive because it was able to utilize the citrate within the medium. And the purpose of this test, of course, is to differentiate between bacteria that are able to use the citrate versus those that cannot. All right, so here is the results table. As I mentioned before, if there is color change, then yes, it is positive. However, if there is no color change, it's still a little bit up in the air, and that is why you have to confirm by checking for growth. And of course, if there is no growth and no color change, then it is officially a negative result. However, if there is no color change, but you do notice growth, then it's still considered positive. And this happens mostly because the medium in itself was not prepped properly. Perhaps the starting pH was a little too low. And so even though the organism was growing and producing a bunch of these nitrogenous compounds that are basic, it just wasn't enough to budge the pH past a certain point that would then activate the bromthiamyl blue indicator within the medium itself. All right, so now that we've kind of gone over the execution and the procedural side of the test, let's kind of dive into the theory and application. So the Simmons citrate test is used to determine the ability of an organism to use citrate as its sole source of carbon. So how does that work? What's going on here? So in the citrate medium in itself, the only source of carbon is sodium citrate. The medium in itself has been controlled so that there was no other source of carbon that bacteria can utilize to produce energy and grow. And so it really limits it to the bacteria that have this specific enzyme known as citrase. And without that, they will not be able to survive. Of course, citrate is part of a series of tests known as INVIC. You will have already performed the methyl red and the VP test, if that sounds familiar to you. The citrate test is part of this family or category known as INVIC that is utilized to differentiate the many species that are found in the genus Enterobacteriaceae. It's a big genus, and there are a lot of species of bacteria in there that are harmful to us humans. And so this test really helps us to kind of separate the species within that family, and of course to kind of separate them from other potential gram-negative rods that belong to different genuses. Now, the citrate test also falls under the category of nutrient utilization tests, and These tests, unlike many of the other tests that you have dealt with, they're they're called they are a highly defined test. Many other tests are just considered undefined, in the sense that they use materials or compounds that are not as granular in a sense. So one good example is a nutrient or a test that is highly defined. For example, our citrate test. There is only a specific chemical, the sodium citrate, that the bacteria can use. However, for an undefined medium, perhaps they'll just use glucose, which is just very generalized, and the majority of bacteria are able to then utilize that. Another example could be to compare using just a specific amino acid versus beef peptone, which is a complex of several different kinds of amino acids. And like I mentioned before, this is very important because this is its way of kind of separating bacteria, almost in the sense of the McConkey agar test that you've done in the past that utilize high salt concentration, as a way to select against certain types of bacteria and to allow one type to proliferate. And just like I mentioned here, of course, the sodium citrate, once again, being the sole carbon source, and ammonium dihydrogen phosphate, which is the sole nitrogen source, as of course bacteria require nitrogen as well in order to grow and survive. Finally, within the citrate test, it contains an indicator known as bromthiamyl blue. And this is important because it allows us to visually see whether or not growth was successful. And just like I mentioned earlier, the bacteria that is able to grow here will also utilize this ammonium dihydrogen phosphate. And as it utilizes it, it will then produce a bunch of these nitrogenous compounds, such as ammonia, that are very basic. And as that stuff builds up, the pH will then rise. And as the pH rises, once it hits a certain point, the bromthiamyl blue indicator is activated, and it leads to this blue color change here. And so the citrate test starts at a pH around 6.9, and when the pH rises to a point of 7.6, that is usually when the indicator will then change colors. Alright, so let's do a quick review of everything we've just gone over. These are the three main points that I believe are important to take away from the citrate test. Once again, the test is used to distinguish between bacteria that are citrase positive versus those that are citrase negative. Citrase being the enzyme that allows the bacteria to use citrate in the medium in order to grow and survive. The test itself is used in order to separate members that are within the genus Enterobacteriaceae, and of course to kind of differentiate them from other gram-negative rods as well. And finally, the blue color change is of course considered positive, but always make sure to check the slants that did not change color, that have remained green, for growth. And if there is growth detected, it is still considered positive even though no color change has occurred. Alright, that is the end of our video. I hope you guys really enjoyed that and you found it helpful. The next video that I will be making, I will be going over exercise 5-8. And if you guys like this video, please like it and leave me a comment. I'm open for suggestions, anything that will help make these videos easier to understand. All of that I would appreciate and is super helpful for me. So thank you guys and have a good night.